Microphone in place, good. Okay, so we're going to talk about glycolysis. So look at the word here, Glycolysis, glycolysis. Now, we talked about hydrolysis before. Remember we talked about breaking the water? Well, lysis here is, think of this as break. Okay, so we're going to break something apart. Glyco, what do you think when you think of glyco? Glycogen, glucose, right? Okay, well here this is going to be for glucose. All right, so we're going to do a process that essentially breaks this apart. And when we break it apart, we're going to release compounds that have energy. Those compounds are going to be ATP, NADH, and at the end, pyruvate. Now, again, why are we doing this? Glycolysis has a lot of energy in it. This thing is full of energy. All right? But it's not in a form that we can use. We can't use the glucose to drive our muscles. We can't use it to build gradients for neurons. We can't use it for a lot of things. So we need to extract the energy from that glucose. And glycolysis is one of the first steps to do that. Now, before we start talking about glycolysis, a few key points. This does not use oxygen. Okay, you're going to look in your book, you're going to look on your lecture notes, whatever. You're not going to see any use of oxygen for glycolysis. Another point to bring up is where it happens. This happens in the cytoplasm. All right, so think about a cell, for instance, right? We have a nucleus with our DNA. We've got the rough endoplasmic reticulum. We've got some smooth ER. We've got a Golgi body. And, we've got multiple mitochondria, okay? This process will take place in the cytoplasm. All right, it happens in the cytoplasm. Now, the results of this, specifically the NADH made, that will be transported to the mitochondria for use in oxidative phosphorylation. More on that later. Okay, so that is a primer on glycolysis. Now let's talk about some specifics about it. Okay, so we're going to start this with glucose. So I'm going to draw glucose up here. Now, one misnomer about glycolysis is it just uses glucose exclusively. Actually, it will use glucose, but at different parts, different carbon-containing compounds can enter. For example, the glycerol from a triglyceride can enter during the glycolysis process. So I don't want to leave you with the idea that this is strictly a glucose-centric model. Other compounds can enter. So what I'm going to do is I'm going to draw glucose, and I'm going to draw it with 6 round little balls here. And these are going to represent my carbons. And this is a fairly common way of representing glycolysis and discussing it in introductory biology because we want to follow the carbons. Okay, so the first step of this, and we want to make ATP, right? But kind of counterintuitive to that is we're going to invest some ATP here. Okay, so what we did is we invested 2 ATPs. And by doing that, the ATP here transferred one of its phosphates, and that's the yellow model right there, to one part of the glucose. And another ATP transferred one of its phosphates, the PO4 group, to the other side of the glucose. Now, this is important because, well, 2 reasons. One is we are inputting energy into this molecule. Now, I said before that glycolysis is going to make us a little bit of ATP, right? But you might think, well, why are we putting energy into it? Well, if you've ever heard the term, you got to make money to spend money, well, that applies on a cellular level as well. We've got to invest a little energy in order to make some more energy. And what this is going to do is these are highly negative. And you remember the structure of these from your first chapter, your first test. It's PO4. And what it's going to do is it's going to destabilize this central bond. Now, I'm not saying that these phosphates are breaking that bond, but it's going to make it easier for an enzyme to break this middle bond. Okay, it's making an easier job for the enzyme to do that. When it does that, it's going to form 2 3-carbon intermediates that we call pGal. Okay, so we have formed pGal in this situation. This is going to be an intermediate. An intermediate here is it's one step on a pathway. So this is going to be an intermediate. Now, next what we're going to do is we are going to pull off some high-powered electrons. So we have something called NAD+. I'm going to draw NAD+ over here. Okay. Now, in class, we liken this to a keg, right? You need to carry something from one area to another area. A keg, for instance, you're going to carry beer from one location to another location, right? You want to use it in another location, but it's located in some other place. So you're going to use a refillable container to carry it from one area to another area, right? Okay. Well, NAD+ is going to be that carrier. That NAD+ will take some electrons, and we say it's reduced in this situation, and it will make NADH. Okay, another one done here. Okay. These NADHs are full. Okay, these are going to carry some electrons to the mitochondria for us so we can make some ATP. Now, we'll talk about that in later on, but right now we're going to say ATP later. Okay, we'll talk about how that makes ATP in a few more sections, but I'll tell you right now, preview of coming attractions, this NADH will make ATP later on. It's not going to make it now, but it's going to make it later on for us, okay? Again, we'll talk about that when it becomes important. So now we have our pGal intermediate that has just been oxidized. The NAD+ has been reduced consequently. And after we've removed these electrons, we are going to do another process. We are going to make 4 more ATP. So you see how I've split this pathway into 2 sections? Because our 6-carbon glucose up here was split, glycolysis, we have formed 2 3-carbon intermediates. So we're going to keep this split going forward. Okay. So now we form 2 3-carbon compounds that we call pyruvate. Sometimes you'll see pyruvic acid. They are not technically the same. There's a slight difference, But for the purposes of this class, pyruvate and pyruvic acid are essentially the same compound. And as we did this, we made, guess what we made? Well, we made ourselves some ATP, and we made ourselves another ATP. Over here, we made an ATP. And over here, we made an ATP. Now, do you remember I said that we had to spend money to make money? Well, we had to spend or invest 2 ATP up here. Remember we had to add those phosphates to our glucose initially? And through this series of reactions, we have now made 1, 2, 3, 4. So we have made 4 ATP right here. Okay, so this process has made us a little bit of ATP. It has also made us pyruvate. Now the pyruvate here and the pyruvate here, these still have a lot of energy in them. Now the energy in pyruvate, 1-pyruvate, is not as much as in glucose. But what we can do is we can take this pyruvate to another step, and we're going to talk about that momentarily. And we can take that and unlock the energy in that pyruvate. Okay? Also, the NADH. We're going to take this, well, we have 2 options really. One is we're going to take it to the mitochondria, and in the mitochondria we can unlock some ATP. 1 NADH is equivalent to approximately 3 ATP. Now we'll talk about how that conversion happens when we talk about the electron transport chain. But for now, we're just going to say an NADH has been made. It's a coupon, if you will, for ATP. Now, in times when there's not oxygen, and we're talking about fermentation here, that NADH can also do another pathway. And again, we will talk about that pathway later on. But the key points I want to go over for this glycolysis. Key point 1 is we started with glucose, has 6 carbons. We then added 2 phosphates, and that was an investment of 2 ATP. It helped to weaken the central bond and allowed an enzyme to break that bond into 2 3-carbon compounds called pGal. These 2 3-carbon compounds were then oxidized, and we made 2 NADHs, one being here and one being here. And we also made 4 ATPs, 1, 2, 3, 4. We also made 2 pyruvates, 1 and 2. Now, the NADH here and here, we can, in some instances, take it to a process called the electron transport chain and make ATP later on. We will cover that in another section. In some other cases, we can take this and enter into a fermentation reaction. Okay, we'll talk about fermentation reactions later. But there are a few choices, if you will, for our NADH. Finally, we have these 2 pyruvates here. Each of these have a lot of energy in them still yet to be realized, to be recovered. What we're going to do with these is the Krebs cycle. And with that, we can then extract more of that energy. Okay, so that's going to wrap us up for glycolysis. But just ask yourself, what is the whole point of this chapter? The whole point of this chapter is to take one form of macromolecule, for example, glucose, and we need to put it into a form of energy we can use. There is lots and lots and lots of energy in glucose, a massive amount of energy, but it's simply not in a form we can use directly. So we do this metabolic process to get some energy that we can use, and that is our 2 net ATPs, because we can use these ATPs to run pumps, to move our muscles, do all sorts of things in the body. And this is also a critical step in converting glucose into other forms that can have even more conversions done to it to release even more ATP. Okay, so that's going to wrap us up for our brief review of glycolysis. And again, we covered a lot more of this in lecture, but this is a good primer, a good review on the process. So we talked a lot about some processes going on with glycolysis. I want to make sure you have an idea of the counts going on here. Okay, so glucose, right? We started this with glucose. How many carbons did glucose have? It had 6 carbons, right? 1, 2, 3, 4, 5, and 6. So each of the spheres you see here represent a carbon. So glucose had 6 carbons. And we added 2 ATPs. And in the process of doing this, we made 2 3-carbon compounds. Okay, so I'm drawing the carbons now. Now we added 2 ATP to do this. Okay, so here we have an input of ATP. I'm going to draw it here. That added a phosphate and another ATP. And that has another phosphate here. So we used 2 ATP. So 2 ATP were used. Now we continued this process on, and after several enzymatic reactions, we formed 2 3-carbon compounds. And each of these 3-carbon compounds we had a special name for, and that was pyruvate. Additionally, we did something else. We made NADH. So we started with this and we made NADH here. And we made another NADH here. Okay. So we made 2 NADHs. And I'll put a little apostrophe because it's plural. So we made 2 NADHs. Now, as we went through this process, we also made 2 ATPs per 3-carbon compound. So we made 4 ATPs. I'm going to put a parentheses gross, right? Your gross salary is what you make before you pay what you owe, right? So finally, again, as I said before, we made 2 pyruvates. Okay, so let's go over what was made and what was used. Glucoses. 1 glucose was used. Okay, so 1 glucose was used in this process. All right. 2 ATPs were used, but 4 ATPs were made. Now, think about your bills and you get your check, right? You have what you make as your gross, and then you pay the taxes, and then you have your net, right? So if I made 4 ATP, but I had to invest 2 ATP, 4 minus 2 equals, well, is 2. So my net ATP My take home is 2 ATP. During the process, I also made 2 NADHs. Now, these become important later on. We can, given the presence of oxygen in the mitochondria, we can make some ATPs with these. Right now, we're not going to count it as ATP, but I'm going to tell you, preview of coming attractions, we can make ATPs with those. And finally, we had 2 pyruvates at the end. Okay, these still have energy in them. We need to unlock the energy in these pyruvates. Now, the next steps after this, the Krebs cycle and oxidative phosphorylation, we're essentially going to do that. Okay, so we're going to see these pyruvates in a future reaction. Okay, so just know the counts here. 1 glucose used, 2 net ATPs made, because we used 2 ATPs, but we made 4. We also made 2 NADHs and 2 pyruvates, each containing 3 carbons.